Sorry for being a little bit late. I'm not sure the network's a little slower today. I was having a hard time logging in. Normally I do that beforehand, but today I thought, oh, I got 5 minutes. It'll be enough time. Wasn't enough time today. The network was really slow. All right, before we begin, can someone first confirm you can hear me?
I can hear you.
Thank you. Are there any questions before we begin?
Quick question. Last Thursday's lecture, so the lecture and labs are separate videos that you post on YouTube. So if I'm gonna look for last Thursday's lecture, it'll be like four videos, or there's
Sorry?
Tuesdays. So I missed the first 25 minutes of last Thursday's lecture because of
Uh-huh.
some things that I legally have to do. So uh it would be like three videos back, right? Because you posted two more videos since last Thursday. You posted Tuesday's videos, right?
I definitely had a lab and a lecture on Tuesday. I don't remember last week if I had a lab or not. Let me share my screen here.
It was the first 25 minutes of the lecture that I missed.
Uh-huh.
So,
So you need to go back and see it. Right. So just find the date. They're
oh, they're dated.
by date. And of course, there'll be lecture and lab in the name as well. Let me see if I can find that. There it is. Can you see my screen?
Yes.
All right, great. No, like I said, you have to hit this link there because it won't open in Canvas. And then it'll open outside of Canvas. There is my website. And so you're talking about maybe this one here on,
It was I when I got home, you were talking about protein denaturing or something, or enzyme
no, I'm
denaturing.
saying I think you want February 5th lecture if you said not last
Oh, the
time,
dates
two
are
times ago.
there.
Yeah, it said the date is on it.
Oh,
And then
but
it
the
says
dates
micro
just kind
lecture and
of
micro lab.
it's just backwards.
Well,
Okay.
it's year, day, and month.
Oh, okay.
Excuse me, that's month and day. Sorry, I got it. It is backwards. Yes. All right. All right, let's get out of here. Guess that's it. I can close that down too. Close that down because I don't use that. Um, all right. Even getting to my Zoom page took me a long time. Like I said, for some reason the network's really slow today. Or maybe it's my computer. I don't know why my computer would be slow. I don't have anything open other than what I have for the class. All right, so let's go to the syllabus. Take a look at the schedule. Today is February 12th, and we should be starting chapter 6. I don't think we finished chapter 5. Did we finish chapter 5? Anyone know?
I think we're like near the end.
Yeah, we're near the end, but I don't think we're at the end. I think we need to finish chapter 5. Oops, I just made that small. Trying to get that out of the way. So I have that we had talked about the integration of metabolism when we talked about amphibolic pathways, where the pathway can go both ways. And as you can see in the digestion of glucose and aerobic respiration, the entire pathway really is reversible. And for those of us couch potatoes, even if we eat fat or a protein, let me see if I can blow that up. And then it breaks down to this amino acid, oxalo, which will be converted to oxaloacetic acid. For those couch potatoes like me, you'll build up glucose. And then when you need ATP production, you'll burn glucose in the normal way. So even if you eat protein or high lipid diet, your body will make glucose and then send it down as glucose. Now, admittedly, if you eat anything, too much of it, your body's not going to store it down as glucose, glycogen. You will eventually convert it to lipids, and then your body will store it down as fat. All right, let me end the show here. Bring this to the top. We're almost at the end here. Photosynthesis. Let's talk briefly about photosynthesis. This is the conversion of light energy into chemical energy. The chemical energy may be used to convert atmospheric CO2 to glucose. We call this carbon fixation. Photosynthesis is performed by cyanobacteria, algae, and everybody knows by green plants. So there are bacteria, there's more than just cyanobacteria. There are bacteria that can engage in photosynthesis. In fact, if I remember correctly, now admittedly that this is mainly the total number of bacteria, not necessarily the total number of species, but most bacteria are either photosynthetic, chemosynthetic, or detritivores, meaning they'll decompose things. So most bacteria are actually that. There's very few that are actually human pathogens. And I don't know the breakup exactly, but most bacteria are photosynthetic, chemosynthetic, or decomposer. Anyways, photosynthesis is a mechanism for synthesizing organic compounds, and these organic compounds are required by animals, microbes, and other organisms which cannot engage in photosynthesis or chemosynthesis and make their own food. And the reason why we can survive is because of some photosynthetic organism fixing carbon to make glucose or some other food ingredient. And even if you say, oh, I'm a carnivore, I eat cows, well, where do you think the cow gets its food from? It gets it from grass, from photosynthesis. Any question about any of that? And all living things have to be dependent on either photosynthesis or chemosynthesis. For most ecosystems on the planet, living things are primarily dependent on photosynthesis, but there are deep sea vents where the life forms are dependent on chemosynthesis. So there are some ecosystems on the Earth where chemosynthesis is the major player. You don't need to know that, I'm just informing you. Let's get back to photosynthesis. It only occurs in photosynthetic organisms. These organisms contain a light trapping pigment. You know one of them, chlorophyll, is a light trapping pigment. When light hits chlorophyll, it causes electrons to move from the chlorophyll, and that light energy can be converted into chemical energy via an electron transport chain. ATP and NADPH are generated. NADPH is similar to NADH in aerobic respiration. It's just that for whatever reason, photosynthetic organisms don't use NADH, they use NADPH. Oftentimes, the NADPH and the ATP are used up to generate glucose, and that we talked about. You generate glucose from CO2 and water, and we call that carbon fixation. That doesn't always have to happen. You do not have to use up the ATP and the NADPH to make glucose. Depends on what the green plant cell or green photosynthetic cell or other photosynthetic cell needs. There are two types of photosynthesis, cyclic photosynthesis and non-cyclic photosynthesis. In Biology 160, or your preliminary biology class, when they talked about photosynthesis, they probably only talked about non-cyclic photosynthesis. Making oxygen, where 6 CO2 plus 12 water molecules in the presence of light make glucose, C6H12O6, 6 oxygen, atoms, or excuse me, 6 oxygen molecules, and then 6 water. Sometimes you can have the water removed from this side, and then you remove that by 6. But I prefer this equation because all of these 12 oxygen atoms come from the oxygen and water. So you need 12 here to get 12 there. Anyways, there is another form of non-cyclic photosynthesis. It does not generate oxygen, so it's anaerobic photosynthesis. And it's this one here. You don't need to know that because I'm not going to test you on it. You do need to know the other two, the non-cyclic photosynthesis generating oxygen. All green plants can engage in this photosynthesis, as well as cyanobacteria, the photosynthetic bacteria I'm going to talk about. Green plants, as well as all other photosynthetic organisms, can also engage in cyclic photosynthesis. Cyclic photosynthesis makes ATP, and that's all it makes. It does not make glucose. The summary reaction of cyclic photosynthesis is you combine ADP and phosphate to make ATP. It depends on what the cell needs. If it needs ATP, even your green plant cell, like, I don't know, green plant cell of a corn plant, if the cell needs ATP, it will engage in cyclic photosynthesis. If the green plant cell needs glucose, it will engage in non-cyclic photosynthesis. So it does depend on what the plant needs. Any question about any of that? All right, let's talk a little bit more about cyclic photosynthesis. It's often called cyclic phosphorylation because you're phosphorylating ADP to make ATP. Light will hit the photosystem 1, which contains the chloroplast in it, causing an electron to move out of the chlorophyll, which in a green plant is inside a chloroplast. But the electron moves out of the chlorophyll. It'll move through this protein, which is a protein in a membrane, move through that protein, which is a protein in a membrane, move through that protein, which is a protein in a membrane, and then move back to this protein, which is a protein in a membrane. And that's why it's called cyclic photosynthesis. The electron moves in a cycle. It moves out of chlorophyll and then moves back to chlorophyll. And that's why it's called cyclic photophosphorylation, because the electron is moving in a circle. In the process, when the electron is moving through this protein, it gives the protein the energy it needs to pump hydrogen ions from this side of the membrane to that side of the membrane. This creates a twofold hydrogen gradient. These hydrogens are going to want to move down to this side of the membrane because there's more hydrogen on this side than that side. There's more positive charges on this side than that side. And so just by the laws of thermodynamics, these hydrogen ions want to move to this side of the membrane. To do so, they can only move through this channel protein. And when the hydrogen ion moves through this channel protein, it gives the protein the energy it needs to make ATP, meaning phosphorylate ADP to make ATP. And this should be familiar to you because it's very similar to the way aerobic respiration makes ATP. The difference is it's more than one channel protein pumping hydrogen from this side to that side in aerobic respiration. It's like right here only. And it makes a twofold hydrogen gradient, which then wants to move to this side of the membrane, making ATP. Any question about any of that? So the hydrogen ions, that's a twofold gradient potential energy. This potential energy is converted into the energy of ATP. And that's another way to make ATP. We did mention that photosynthesis can make ATP. Any question about any of that? All right, the last slide of this chapter. Let me move that. Requirements for making ATP production. Oh gosh, bring that up there. The energy source can vary for photosynthesis. The electron comes from actually chlorophyll, and in cyclic photosynthesis, the electron travels back to chlorophyll. And in animals, glucose will be used. The in aerobic respiration, glycolysis can make some ATP, but most of it is the energy will be taken by a carrier, electron carriers like NAD plus, NADP in plus in green plants. And then the final electron acceptor for aerobic respiration will be oxygen. For anaerobic respiration, it'll be a molecule other than oxygen, the final electron acceptor. In fermentation, it'll be an organic chemical compound like pyruvate. And not listed on this slide, but for photosynthesis, at least in cyclic photosynthesis, the final electron acceptor is chlorophyll, the same molecule that the electron came from. Any question about any of that? The point being the energy source can vary, the electron donor, and then the electron acceptor can also vary. Any questions about chapter 5? All right, I don't know if I saved that, so let me save that. Close that down, and now we're supposed to go to chapter 6. All right, this slide is showing you the major goals and a rough outline. Know the terms. What's a psychrophile, a psychotroph, a mesophile, a thermophile, a hypothermophile, an acetophile, a halophile. What's an aerotolerant anaerobe? What's a facultative anaerobe? What is binary fission? What are reproductive spores? And what are endospores? Remember, we've talked about endospores at least one time already, so this will be the second time. We will talk about endospores at least 3 times in this class. 2, be able to understand the various requirements that cells have for growth. 3, understand the 4 phases of growth. And then 4, compare the different methods we have for measuring microbial growth. Any question about any of this? When we're talking about microbial growth, we're referring to an increase in the number of size of the number of cells. We're not talking about the length of the cell growing. I used to teach middle school students, and obviously when we're talking about them growing, hopefully we're not talking about them reproducing. Any question about any of that?
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in 2. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
Any question about any of that? There are requirements for microbial growth, and actually there are requirements for any organism's growth. They do have to have physical requirements and chemical requirements. physical requirements, the microbe has to be at the correct temperature, the correct pH, the correct osmotic pressure, or the correct salinity is another way of wording that, for it to grow. If it's not in the correct environment, and if it's way out, it will not grow at all. If it's slightly off, like slightly the wrong temperature, it can grow at reduced speed, but it can grow if the temperature is slightly off. And then if it's way off, it will not grow. Microbes also have a chemical source. They must have a source of obtaining carbon, a source of nitrogen, a source of sulfur, a source of phosphorus, a source of trace elements, a source of oxygen, or if they are an anaerobe, they have to have no oxygen present in order to grow, and a source of organic growth factors if they require them. Any question about any of that? No, it does depend on the cell for what it does require. Like something which is a oxytroph, meaning it can get its own carbon from CO2 in the air. That's the only source of carbon it needs. Doesn't need another source of carbon. And if it can fix nitrogen, pull nitrogen down from the air, that's the only source of nitrogen it needs. So these carbons and nitrogens do not need to be supplied in the media of an oxytroph or a nitrogen fixing bacteria. But you still would need to supply a source of sulfur, phosphorus, trace elements, which all organisms need those, oxygen if it requires it. Most oxytrophs will not need an organic growth factor, but something like Streptococcus pyogenes requires an organic growth factor to grow, and that's why it will not grow on a general purpose media. You can grow Streptococcus pyogenes on blood agar because the red blood cells require the two organic growth factors necessary for Streptococcus pyogenes to grow. Any question about any of that? Trace elements can often be obtained from organisms just in the water or from their environment. Like actually most of the trace elements that humans require, we get from the food we eat and the water we drink. For those of you who want to know, we don't have enough of iodine in Western Oregon and Western Washington. So if you were to only eat food grown in Western Oregon or Western Washington, you would need to supplement your diet with iodine. And you can get that from iodized salt or something that's made with iodized salt. And then nowadays, food, especially in the grocery store, you don't know where it's come from. A lot of it's not grown in Western Oregon or Western Washington. And the reason why we need iodine is because the rainwater has washed out iodine from our soil. That wouldn't be true for something grown in Eastern Washington or Eastern Oregon. But in Western Oregon or Western Washington, you do need iodine in your diet, or you need to buy food that's not grown in Oregon. Like Brazil nuts, they don't grow in Oregon and Washington. Any question about any of that? All right. So all organisms have a physical requirement of the correct temperature. Each species has its minimum, maximum, and optimum growth temperature for what it'll grow at. Optimum temperature will be the temperature the bacteria or microbe can grow at the fastest rate. The minimum temperature will be the lowest temperature the organism can grow at, and the maximum will be the highest temperature the organism can grow at. You can classify microorganisms into the groups of their preferred temperature range. Let's go ahead and go to the next graph. We have psychrophiles, and these organisms grow best around 10 degrees Celsius. That's the temperature of the refrigerator. their range being something like minus 10 degrees Celsius to 20 degrees Celsius. Beyond that, they do not grow because they like cool temperatures. How they can grow below zero is they make their own antifreeze. Not all psychrophiles make antifreeze, but some do. And so some of them can grow down to a minus 10 degrees Celsius. Psychotrophs are organisms that grow best around room temperature, 20 to 23, 25 degrees Celsius. These are most organisms that grow, well, I guess in your house or in the soil or even in the ocean. Most of them are psychotrophs. Mesophiles, organisms that grow best around 37 degrees Celsius. They stop growing close to around 50 degrees Celsius. And also the low temperature would be about 10 degrees Celsius. All human pathogens are mesophiles. So this group you do need to know because all human pathogens are mesophiles. Any question about any of that? And that's because they grow at temperatures around our body temperature. Now, let me warn you that this graph is not rigidly defined. We do have mesophiles that grow on our skin, and their optimum temperature for growing is actually around 30 degrees, which is about the temperature of the skin. They do not have their optimal temperature growth at 37 degrees because the skin is around 30 degrees. We still call them mesophiles because it's close enough to 37 degrees. My point is that the exact optimum is not rigidly defined, and then the range is obviously not rigidly defined. Different species will have different range, meaning there are different minimal and different maximum temperature they can grow at. Any question about any of that? All right, we also have thermophiles. Their optimum is around 60 to 65 degrees Celsius that they can grow at. Hypothermophiles was a category of microorganism that wasn't even known when I took microbiology. These organisms grow best 90 degrees or higher temperature. Remember, 95 degrees Celsius is about the average for the hypothermophile, and 100 degrees Celsius is the temperature that water will boil at least at sea level. And these hypothermophiles can actually grow that at that temperature. There are some families in Mexico that have a volcanic vent, and they'll actually throw their corn in a gunny sack and then throw it in the volcanic vent or the water coming out of the volcanic vent to cook their corn and then take it home to eat it. In that water are lots of hypothermophiles, and these people are obviously eating hypothermophiles. The hypothermophiles do not make them sick. Obviously, the hypothermophiles cannot grow at 37 degrees, so we wouldn't expect them to be a human pathogen. But these hypothermophiles are all appear to be non-toxic. Don't seem to cause any trouble to humans. Any question about any of that? That isn't true for the psychrophiles, the organisms growing in the refrigerator. As you know, they can cause food spoilage and then contaminate the food, making the food unedible to eat. And that usually happens about a week after the food has been in the refrigerator. The psychrophiles can grow, psychrophiles can grow to a temperature high enough to contaminate the food and then cause food spoilage. So unless the food has some other preservative in it, like either acid or salt or sugar, the food in the refrigerator should be tossed about after 1 week it's been in the refrigerator because the psychrophiles can grow in the refrigerator, causing food spoilage of the organisms. Any question about any of that?
I
The
just have a
thermophiles can actually grow in the rotisserie, like you see the chicken spinning in the rotisserie at Safeway or some other grocery store. Those organisms can actually grow in that rotisserie. So if the chicken looks like it's been there too long, looks like it's too old, it's probably better not to eat it. Although obviously the chicken cannot be in the rotisserie for a week, which is, well, let me think about that. That would take less time than a week. I'm not sure how quickly the thermophiles could grow. I was saying that the chicken wouldn't be there for a week, but actually the thermophiles will grow more rapidly than organisms in a refrigerator. So I'm not sure how long they would grow until they became toxic level. But my point is, if the chicken looks like it's been there for too long, you're probably best not eat that chicken. We talked a little bit about food spoilage. Let's talk about it in a little more detail here. The danger zone of food spoiling is about 15 degrees Celsius or 60 degrees Fahrenheit to 52 degrees Celsius. That would be like 130 degrees Fahrenheit. At this temperature, bacteria can grow rapidly. Some bacteria can produce toxins, making the food not edible after a little more than 4 hours. The bacteria can grow at a more wide temperature range, like, I don't know, 7 degrees Celsius. That's about 40 degrees, 41 degrees Fahrenheit to 50, maybe 60 degrees Celsius, which would be
35 degrees Fahrenheit. I just retook my food handler's card test last night, so I went over all of this.
Okay. The point is that the bacteria can grow outside of this range. This is the rapid growth range where bacteria can grow slower at either a little lower temperatures or a little higher temperature. And so you can get bacterial growth over a longer period of time. And if the food is at this temperature for too long, it can cause spoilage, and you don't want to eat the food like in this range after the food is sat out for a little over 4 hours. This is also a problem when you refrigerate food. If you refrigerate food and it's in a small container, the food will quickly get through the temperature range the bacteria can grow at, in which case the bacteria cannot grow to elevated numbers and then cause the food to spoil. But if you refrigerate in a very large vat, the food will cool too slowly, and the food will be in this temperature range long enough for the food to contaminate the bacteria to contaminate the food. This is particularly a problem with rice, which tends to have endospores of Bacillus cereus, which Bacillus cereus can cause the food to spoil. And if someone were to cook up rice and then put it in a very large vat, put it in the refrigerator, the food would spoil because it would be in this temperature range for 1, 2, 3, 4, 5, greater than 6 hours. Is that right? 3 to 8, I guess that's 5 hours. 1, 2, 3, 4. Yeah, greater than 5 hours. And what you need to do is just split up the big vat into smaller vats, and then the smaller vat will cool quickly enough so that the temperature range will not be long enough for the food to have the spoilage microorganisms grow in them, and then potentially cause a toxin. Any question about any of that? Most bacteria also have a pH requirement. Most bacteria grow well between around pH 7, so something like between pH 6 and 8. This is actually a problem when you're growing microbes in a microbiology lab, because as the microbes grow, they make acidic products. The acids can then inhibit the growth. To minimize this acid buildup, inhibiting the growth of microbes, we tend to put a pH buffer in the culture media. And then the buffer will minimize the acid buildup. So this is true of bacteria. Most molds and yeast actually grow at a slightly acidic temperature, and they actually grow well between pH 5 and pH 6. So slightly acidic. Remember, your skin is about pH 4, your stomach, about pH 1.5, at least when it's producing acid. Acidophiles can grow in acidic environments, such as a volcanic environment, where the pH will be between pH 0 and pH 3. So the acidophiles can grow in very acidic environments. Any question about any of this?
What was the pH range that was optimal for bacteria again?
Most bacteria grow between pH 6 and 8, meaning they need to be around pH 7. It's not true of a bacteria, which is an acidophile, but most bacteria only grow about pH 6 to 8. After that, they stop growing. Oops. I guess I am here. All organisms have a physical requirement that they have to grow in the correct osmotic pressure. All microbes and all living cells actually require water in order to grow. If you have the cell in the incorrect osmotic pressure, the microbe cannot grow. High osmotic pressures can remove necessary water from a cell, such as when you put a microbe in a hypertonic environment. The water will then leave the cell, causing the cell to go through plasmolysis, where the water will leave the cell. The cell membrane will shrink, pull away from the cell wall. And depending on how sensitive the cell is to osmotic pressure, the cell can die. If it's an endospore, well, that's not likely to kill the cell. Plasmolysis will not kill an endospore. But if it's a cell like E. coli, plasmolysis will kill the cell. So you put it in a hypertonic environment, E. coli will die at least within a day, maybe even sooner. It does depend on the cell, like Staphylococcus aureus can survive high osmotic pressure for much longer than E. coli. Staphylococcus aureus can survive high osmotic pressure for at least a week, maybe a few weeks, but eventually even Staphylococcus aureus will die by plasmolysis. A cell prefers to be in isotonic solution where no water enters the cell and no water leaves the cell. Hypertonic solution was having higher solute concentration outside the cell will pull the water out of the cell. Bacteria often find themselves in a hypertonic solution. Even a human pathogen growing on a human, they tend to find themselves in a hypertonic solution. However, the bacteria cell wall can protect the bacteria cell from dying in a hypertonic or a hypotonic solution. And remember, that is how penicillin G actually kills the bacteria. What it does is it puts holes in the cell wall, and then the plasma membrane can protrude out of the hole. Oops, sorry about that. And most bacteria, when it's in us, it's actually in a hypotonic solution, but the cell wall can protect the bacteria. And that's why penicillin G can kill the cell, because the penicillin G puts a hole in the cell wall, and then the water will move into the cell and eventually kill it. High osmotic pressures can be used to preserve food, like salted meats, molasses, honey, salted fish. All of these are being preserved, at least to a degree, by the high osmotic pressure. And as you know, molasses, as well as honey, can stay at room temperature for over a year and it will be fine. And that's because when the microbe falls into it, the water leaves, and most bacteria will die by plasmolysis. It's true an endospore were to fall into the molasses, the endospore will not die. However, it's not going to grow either. And so the bottle of molasses can survive for 20 years and be good. And I know this because my mother had a bottle of molasses that I figured was about 20 years old. And I made molasses candy out of it and gave it to my father, and he enjoyed it. Didn't kill him. So we know that molasses can be 20 years old or older. By the way, that molasses bottle became eventually we decided was at least 30 years old. And my mother said, it's time for me to throw that away. And she and I weren't using it. The last time it had been used was when I made molasses candy for my father 10 years ago. And I said, well, you may as well. We're not using it. So throw it away. Any question about any of that? All right. Go to the next page. Halophiles are organisms that like growing in a salt environment. They grow in high salt concentrations. We have two types. Halo means salt lover, by the way. You don't need to know that, but halophile is something that can tolerate salt. There are two types of halophiles, an obligate halophile and a facultative halophile. An obligate halophile is one that requires high salt concentrations to grow. An example of a salt-loving microbe would be a microbe that lives in the Dead Sea. This is the sea between Jordan and Palestine, where the salt concentration is 30%, and that's as high as you can get, and then the salt precipitates out of the water. Those organisms growing there are all obligate halophiles. A facultative halophile is one that does not require high salt concentrations to grow, but it can tolerate salt concentrations greater than 2%, and some can even grow at 15% salt. An example of a facultative halophile is Staphylococcus aureus. It can grow well on TSA or nutrient agar, which is about isotonic or 0.9%, meaning 0.9% sodium chloride. It can grow well on that plate. And yet it can grow on your skin, which can reach as high as 20% sodium chloride. Depending on how much you're sweating and the last time you had a shower or bath, your skin can reach 20% sodium chloride. Why? It's because when we perspire, we secrete salt, and then when the perspiration evaporates, the salt crystals are left behind. Any question about any of that? All right, so Staphylococcus aureus is a facultative halophile. And I used to know the name of one of the obligate halophiles, but I don't remember the name of it now. If it comes to me, I'll mention it, but you don't need to know it. In a hypotonic solution, low osmotic pressure, distilled water can enter the cell by osmosis, and that may cause the cell to lyse, like a red blood cell which has no cell wall. More and more water will enter the cell. The red blood cell will puff up, sort of like a fat jelly-filled donut, and then eventually the water will move into the cell until the cell membrane bursts, killing the cell. Now, that will happen to a bacteria as well, but most bacteria have a cell wall. The cell membrane will reach the cell wall, and then the cell membrane cannot expand any further. So the cell wall protects the cell membrane, and because the cell membrane cannot expand any further, that means no more water can move into the cell. Why can no more water move into the cell? Because there's no room for the water to move into the cell. And so the cell wall can protect the bacteria cell or the plant cell from osmotic lysis, preventing water from moving into the cell, causing the cell membrane to expand until the cell membrane bursts. Remember, a hypotonic solution is one with low osmotic pressure where there's more water outside the cell than inside. The tonicity terms will be there's fewer solutes molecules outside the cell than inside the cell. Any question about any of that? All right, all living organisms have must have a carbon source. They have to get their carbon from someplace. The typical cell has about half of its dry weight from carbon. So think about that. You take the water out of a cell, half of the weight of that cell will be from carbon. Carbon is used in structural organic molecules, so it's a large part of the cell. Carbon is an energy source for cells. Most cells obtain their energy needs from a carbon molecule, such as glucose. Heterotrophs use an organic carbon source to obtain their carbon. An organic carbon source would be glucose. We are a heterotroph. We get our carbon needs from either glucose or some other food molecule containing carbon, which I think all of the food molecules contain carbon, but so we are a heterotroph. An autotroph, though, can get its carbon from CO2 dissolved either in the air or in the water. Everyone knows a green plant is an autotroph, getting its carbon needs from CO2. But there are green photosynthetic bacteria as well as chemosynthetic bacteria, which are autotrophs, and they can get their carbon from CO2, either dissolved in the air or in the water. Any question about any of that? Nitrogen is another chemical requirement. You don't need to know this, but 14% of the dry weight of a cell is actually nitrogen. You find nitrogen in amino acids, protein, in DNA, RNA, and ATP. Nitrogen can often be obtained by decomposition of proteins. Like humans get most of their nitrogen needs from decomposing proteins in our diet. Bacteria, too, can obtain nitrogen from decomposing protein. Like E. coli, if you have protein in the media, E. coli can get its nitrogen needs from decomposing protein. But many bacteria can also use ammonium, NH4 with 1 plus charge, or nitrite, NO3 with -1 charge, or some other nitrogen source. Like even E. coli can use nitrate. I think it can use ammonium ion as well. But it can get it from protein as well. A few bacteria can even harvest nitrogen and nitrogen in the air, meaning nitrogen gas from the air. And these organisms we call nitrogen fixers because they can pull nitrogen directly out of the air and then make that nitrogen available to other organisms, usually when this bacteria or archaea dies. It will pull nitrogen out of the air. That nitrogen will usually be made into some protein or something in the nitrogen fixing bacteria. And then when that bacteria die, that nitrogen can be available for other organisms to use. Any question about any of that? So it depends on the species. Some bacteria obtain their nitrogen from proteins, decomposition of proteins. Some can get it from ammonia or nitrate, or some other nitrogen source. And a few of them can even get nitrogen from the air. Trying to remember the name of that one. Ah, don't remember the name of that one. Sorry. It's actually in our one of the ones we use for unknown bacteria, but I'm not remembering the name. Oh well. Sulfur is also a chemical requirement. All organisms must obtain sulfur as a source. Sulfur is needed in certain amino acids, like cysteine, methionine. It's also required for thiamine and biotin. These are two B vitamins. And sulfur is required for DNA and RNA synthesis. Sulfur can be obtained from decomposition of proteins. So many organisms, including humans, can obtain sulfur from decomposing proteins. Many bacteria can use sulfate as their sulfur source, or even some bacteria can use hydrogen sulfide gas as their source of sulfur. Hydrogen sulfide gas is toxic to us, so that'd be poisonous to us. And then we cannot get sulfur from sulfate. Any question about any of that? If you want to know the reason why an egg yolk has a particular taste of an egg yolk, it's from the sulfur in the egg yolk. Phosphorus, or all organisms do have a requirement for phosphorus. Phosphorus is required for DNA, RNA, ATP, and then phosphorus is required in our cell membranes. Remember, phospholipid is the main component of the cell membrane, and so we do need phosphorus for our cells. Phosphate is the most important source of phosphorus, and as far as I know, all organisms can use phosphate, including humans. We can get phosphate oftentimes from milk, but other foods we eat as well. And then we can get it from, well, we don't really eat much DNA, RNA, ATP, or membranes, but we can get it from that. Any question on these chemical requirements? All organisms have trace elements as a chemical requirement. Trace elements are inorganic elements required by microbes in small amounts in order to grow. Trace elements can often be an enzyme cofactor. Examples of trace elements include iron, copper, and zinc. Most organisms can get trace elements from the water in their environment, like we get it from the water we drink, or it's in the food that we eat in trace amounts. Iron, obviously, if you eat anything of red meat, anything with blood will have iron in it. So good source. Zinc, I used to know this. Green vegetables can contain zinc.
I thought zinc was antimicrobial.
Uh, it can be, but they also require it in trace elements. So zinc is not um, antimicrobial in trace amounts. You need a certain amount before it became antimicrobial. Copper, too, can be antimicrobial at higher amounts, but copper is required in trace amounts, I think, by all living cells, certainly by microbes and by humans. One source of copper, you can eat raisins and you can get a fairly good source of copper. Any question about any of that? All right. So most organisms get their trace elements just in the water um, they would consume or the food that they consume, including humans. Organisms may have an organic growth factor as a requirement. An organic growth factor is some biological molecule that is required by the organism in order to survive, reproduce, and whatnot, maintain its health. Um, these organic compounds must be obtained from the environment because the organism cannot synthesize it, the organic growth factor itself. Some examples of an organic growth factor would be all vitamins, like vitamin C for humans. We cannot make vitamin C, so we need it as an organic growth factor in our diet. A good source of vitamin C, almost all fruit, but especially citrus fruit, will be a good source of vitamin C. Certain amino acids and fatty acids or other organic growth factors, if it's called an essential amino acid or an essential fatty acid, that is an organic growth factor that we must have. Like the amino acid lysine is an essential amino acid. You don't need to know lysine, you just need to know that you need to have essential amino acids because we cannot make it. So we have to have it in our diet. And generally, you can get lysine just from protein. Some other examples of organic growth factor include purines and pyrimidines. They may be organic growth factors as well. Any question about any of that? All organisms have an oxygen requirement, even if they require the absence of oxygen. That is a requirement for that cell to grow in an environment without oxygen. Other oxygens have a requirement for oxygen. They will not grow unless oxygen is available to them. In a sense, molecular oxygen is a toxic substance because even for aerobes, they can make toxic oxygen metabolites, which are toxic to even the aerobe. Is that clear? So even to humans, which require the presence of oxygen, or else we would die, because we absolutely require oxygen for aerobic respiration. But even in humans, we make four toxic forms of oxygen. It's an oxygen metabolite, which is toxic to us. Unfortunately, our body has means to remove these toxic forms of oxygen, which actually all aerobes do have these means to remove them because they'll make the toxic form of oxygen. They have to have some way of removing it. Before
So
toxic, Go
So
on.
my molecular oxygen is fine, but the metabolites of molecular oxygen are poisonous.
Yes, we can breathe molecular oxygen fine. However, when we use that molecular oxygen, we can make at rare amounts a toxic form of oxygen, a metabolite of oxygen. The first toxic form of oxygen is singlet oxygen. This is oxygen gas, which has an electron boosted to a high energy state. This oxygen is extremely reactive, and this oxygen is actually present in phagocytic white blood cells, which use singlet oxygen to release it on something the white blood cell wants to kill. Singlet oxygen is primarily removed by quenching the antioxidant to something else like a carotenoid, vitamin E or vitamin C, which then removes the singlet oxygen by binding to it. It does remove vitamin C or vitamin E as well, but it's better to remove the vitamin C and then get rid of the singlet oxygen than to have the singlet oxygen around, because the singlet oxygen is highly reactive and it would damage your cell. Any question about any of that? And actually, that's what the white blood cell does. It damages the cell the white blood cell wants to damage by using singlet oxygen, secreting singlet oxygen onto the cell. Another toxic form of oxygen is a superoxide free radical. This is oxygen gas containing an extra electron, that symbol there. Let me blow that up so you can see it a little better. That symbol there with an -1 charge. So this oxygen gas has an extra electron on it. Why it has a -1 charge? And it's a free radical. This is a highly unstable molecule. It is formed during normal aerobic respiration in small amounts by aerobic respiration. Free radicals, especially the superoxide free radical, can steal an electron from any molecule it bumps into, which eventually will then steal an electron from another molecule it bumps into until DNA is bumped into, and then that can mutate the DNA. So the trouble with superoxide free radical around is if it bumps into DNA, it can damage the DNA, or if it bumps into another molecule, it'll steal the electron from that molecule, making that molecule a free radical, and then that molecule can bump into DNA to damage the DNA. Our cell can neutralize superoxide free radicals with the enzyme superoxide dismutase, abbreviated SOD or SOD for S super O oxide D dismutase. What you do is you have two free superoxide radicals that combine with two hydrogen ions in the presence of the enzyme SOD to make hydrogen peroxide and oxygen. Hydrogen peroxide is also toxic to the cell, so the cell has to have some means of handling the hydrogen peroxide if it has the enzyme SOD to neutralize the superoxide free radicals. And we do have another form of oxygen which is toxic, and that is a peroxide anion, shown here. And the most common peroxide anion in the human body is hydrogen peroxide, shown here. Sorry, here. We can remove a peroxide anion with one of two enzymes. Catalase, which can take two hydrogen peroxides and make two water molecules and oxygen. So we have the enzyme catalase, at least humans do, in our cells to remove hydrogen peroxide. Some bacteria have the enzyme peroxidase instead of catalase to take hydrogen peroxide to hydrogen ions to make two water molecules. And a bacteria that has this is a member of the Streptococcus family or the Endococcus family. It has the enzyme peroxidase, and that's why this family of microbe can grow in air. Because in air, peroxide anions are made, hydrogen peroxide is generated. The cell can survive because it makes peroxidase to remove the hydrogen peroxide. And that's why Streptococcus pyogenes can grow in air. Our cells can grow in air because we have the enzyme catalase, as well as many bacteria cells like members of Staphylococcus species like Staphylococcus aureus can grow in the air because they can convert hydrogen peroxide into water and oxygen using the enzyme catalase. Any question about any of that? All right, the fourth and final toxic form of oxygen is the hydroxyl radical. This is the OH hydroxide ion with one free electron. Electrons don't like to be just one. They like to either be two or none. And so this is a oxygen radical. Here it's called the hydroxyl radical. It differs from hydroxide ion, which has a negative charge. And that's because it doesn't have one free electron. It has two. And that's why it has a negative one. It also differs from the alcohol group in a molecule. That doesn't have a free electron. It actually has two electrons, one bonding to that one there, and then one bonding to this hydrogen there. Anyways, hydroxyl radicals are generated in low amounts by aerobic respiration. They can be generated by ionizing radiation, and they can be generated when hydrogen peroxide reacts with a certain metal ion, producing hydroxyl radical. Antioxidants remove this free radical and other free radicals from our cells. Two antioxidants that remove hydroxyl radicals include vitamin E and vitamin C. So vitamin C and vitamin E are antioxidants that can remove the hydroxyl radical. When they do, they remove the vitamin C, but the vitamin C ties up the hydroxyl radical, removing it from the cell. Any question about any of that? All right, where's my chart here? There are organisms that do require oxygen. We call these aerobes. Microbes that use oxygen to produce ATP are called aerobes. Microbes are also classified on the basis of the effect that oxygen has on their growth. Like an anaerobe are organisms that do not require oxygen. And most anaerobes actually do not grow in the presence of oxygen. So we'd say they are obligate anaerobes. You can classify microbes on the basis that the effect of oxygen has on their growth. Let me blow this slide up. So the easiest one to explain probably is this one here, the first one. An obligate aerobe is an organism that grows in the presence of oxygen it requires oxygen, and in the absence of oxygen, there is no growth. You can inoculate a thioglycolate media tube with an arrow, and you'll see growth looking like this, where most of the growth will be at the top of the tube because the thioglycolate tube will have it removes oxygen from the tube, but it creates a gradient of oxygen. On the bottom of the tube, there is no oxygen because the thioglycolate has removed it from the media. But at the top of the tube, we get oxygen coming in. This is an uncapped tube, oxygen coming in. And so the top of the tube has got full oxygen. For an obligate aerobe, we'll see the most growth at the top of the tube because the cell requires oxygen to grow for aerobic respiration. And as you go down the tube, you'll have less and less oxygen. So you'll have less and less growth. So you can put one dot right there, and then that would be the bottom of the tube where there's a little bit of oxygen way down here in the tube. So you have a little bit of growth because there is some oxygen. But further down in the tube, you have no oxygen, and so you have no growth. And so you can think of this as a triangle with the most growth at the top and then the least growth, I don't know, maybe down about halfway down into the tube. Any question about obligate aerobes? The next easiest one to explain is an obligate anaerobes. These are organisms that do not use oxygen to grow, and an obligate anaerobe cannot grow in the presence of oxygen. So what you find is on the bottom of the tube, you find most growth. And as you go up the tube, you'll have less and less growth. So there's 3. Let's make another one here where you have 1. And that's because as you go up the tube, you'll have more and more oxygen. So you'll have less and less growth because these organisms do not like growing around oxygen. Any question about any of that? So if you go over halfway, you have no growth. There's too much oxygen for the organisms to grow. And then down about halfway, you have a tiny bit of growth because there's some areas of the media without oxygen. So you have a little bit of growth. And as you go down the tube, you'll get more and more growth until you reach the bottom of the tube, which will have the most growth because there's no oxygen in the bottom of the tube. And that's an obligate anaerobe. The next easiest to explain is a facultative anaerobe. These organisms grow in both the presence of oxygen as well as the absence of oxygen. In the presence of oxygen, they will use oxygen to make ATP because that's the most efficient way to make ATP. So these organisms will grow best at the top of the tube because at the top of the tube, they can engage in aerobic respiration. And so they will grow faster at the top of the tube than anywhere else of the tube. And they'll have their slowest growth at the bottom of the tube where there is no oxygen. The facultative anaerobe can grow in the absence of oxygen. The facultative anaerobe will grow either with fermentation or with anaerobic respiration in the absence of oxygen. So this one would be, once again, a triangle. The top of the tube, you have the most growth, and then at the very bottom of the tube, you have the least growth. But you have more growth in this tube all the way down to the bottom, whereas this tube, you only go down about halfway. An obligate aerobe. A facultative anaerobe. So this is not an obligate anaerobe because it can grow in the presence of oxygen, and actually it grows best in the presence of oxygen. The other to explain, and easiest to explain, is an aerotolerant anaerobe. These are anaerobes, so they will grow in the absence of oxygen, but they're aerotolerant anaerobes, so they can grow in the presence of oxygen. And these organisms grow uniformly throughout the tube. grows well at the top of the tube as they do on the bottom of the tube. And the reason why they can grow at the top of the tube is they have enzymes and everything else they require to remove the toxic forms of oxygen. And that's why they're an aerotolerant anaerobe. All right, the last to explain is the microaerophiles. These are aerobes, so they do require the presence of oxygen to grow. In the absence of oxygen, look at the bottom of the tube, they do not grow. And they are aerobes because they perform aerobic respiration. However, microaerophiles are organisms that do not grow best at full oxygen in the air, about 21%. They grow best between 2% and 10% oxygen. And so they grow best in the middle of the tube where the oxygen is around 2 to 10% oxygen. And as you go down the tube, you'll have slower growth until the end of the tube. You'll have so slow growth you don't have any growth because it does need oxygen to grow. And as you go down the tube, you'll have a little bit of oxygen. So you get a little bit down here. And then as you go up the tube, you'll have less growth. You get a little bit of growth here at the top of the tube because it can grow in full oxygen. It just doesn't grow best in full oxygen. In an organism which is a microaerophile, let me see if I can remember this. Helicobacter pylori, the bacteria that grows in your stomach and can cause ulcers.
Stomach ulcers.
Yeah, stomach ulcers, is a microaerophile. It grows best in reduced oxygen concentration. The stomach swallows air, so that's why it does have some air, but the microbes in the stomach, some of them can perform aerobic respiration and then remove the oxygen from the air. And that's why Helicobacter pylori grows best in the reduction of oxygen, and that is other microbes can use up that oxygen in the stomach, reducing it down to the optimum temperature, oxygen concentration for Helicobacter pylori, which would be something like 2% oxygen, 2 to 10%. Any question about any of that? All right. Actually, this is a good place to stop. So I'm going to stop here and ask, are there any questions? All right. If there's no questions, let me see if there's a lab today. I don't remember. I didn't mention it earlier. Yes, there is a lab today. So I'll see you at 6:30 in the lab. Bye.